Hi friends, today we'll be talking about what is called as your test for determining qualitatively the presence of a typical reducing sugar matlab in a given sample. So when we are analyzing carbohydrates, as I told, like if you have a sample and you have analyzed that there are certain amount of carbohydrates which are present in it, some kind of, like if you have performed Molish test or if you have performed Anthron test, and you have come to a conclusion that yes, my given sample, it contains some kind of carbohydrate, but I am not sure that whether which kind of carbohydrate is present. Say I also performed what you call as the iodine test to detect that whether there are polysaccharides in that present or not, and then I am now trying to find out that whether the sugars which are present in the given sample, they are reducing or non-reducing sugar. So like the sugars like which under alkaline condition, they have the aldehydic group or the ketonic group, and in these conditions they act as the reducing agents. So those particular sugars are called as the reducing sugar, right? So what is a reducing sugar? A sugar which will act as a reducing agent and which itself will get oxidized. So it will donate some hydrogens, it will donate some electrons, and itself will get oxidized and will act as a reducing agent. That particular sugar is called as a reducing sugar. Like polysaccharides, matlab they come under non-reducing. All your monosaccharides like your glucose or fructose, right, they come under reducing sugars. Remember, all monosaccharides, they are reducing, right? You have disaccharides, some of which may be reducing, some of which may be non-reducing. You may please name some of the disaccharides which are reducing and some of the disaccharides which are non-reducing, right? But remember, but monosaccharides, all of them, they are reducing. So under alkaline conditions, they will donate the hydrogens, they will act as the reducing agent, themselves they will get oxidized. So one simple test is your Benedict's test. So Benedict test, it actually can be used in both ways, qualitatively as well as quantitatively. Like you can find out that whether the sample, it contains the reducing sugar or not, yes. And once I know that it contains the reducing sugar, then I can find out how much amount of that reducing sugar is done. Yes, but the reagent used are different. Let us first today talk about the qualitative Benedict's test, which helps us to differentiate the reducing and the non-reducing sugar. So remember, Benedict's test for estimating or for detecting the reducing sugar. You use sodium citrate here, which acts as a chelating agent. So it will chelate the cupric ions there so that they don't form a precipitate. So otherwise what will happen is the cupric ions, they will get precipitated in the alkaline condition themselves itself. So sodium citrate, it will act as your chelating agent here. If the reducing sugars, they are present, they will form what is called as a brick red precipitate. Now there are series of color changes. Matlab, the Felling's, the Benedict's reagent rather which you prepare is somewhat like this. It will be bluish in color. After the reaction, if there is presence of reducing sugar, these particular, depending on the type of the sugar or the amount of the sugar, you will get different colors. Like you may get a green color, you may get an orange color, you may get a brick red precipitate if there is a huge quantity of this reducing sugar which is present in the given sample. Yeah. So if you look at the series of reactions which take place, your sodium carbonate, it reacts with water to form sodium hydroxide, which will react with copper sulfate to form cupric hydroxide. This cupric hydroxide will dissociate to form what is called as your cupric oxide. And this particular cupric oxide now, as you can see, in presence of the reducing sugar, your reducing sugar, as I told, itself will get oxidized. So the carbonyl group, it may be an aldehydic or a ketonic group, the CHO or C double bond O kind of group. The carbonyl group which is there, that will be converted to a carboxylic group. So your glucose will be converted to your gluconic acid, and your cupric oxide will be converted to your cuprous oxide, which forms different colored precipitate. Here you can see. So there is formation of this brick red precipitate, indicating that there is a huge amount of reducing sugar present in the given sample. How do I prepare the reagent? Like if I am preparing like 1 liter of the reagent, here are the calculations. You can take 173 grams of sodium citrate. To add, you can add 100 grams of anhydrous sodium carbonate. You mix these two in 600 ml of your hot water and dilute it and make the volume to 800 ml. To prepare the second one, you have 17.3 grams of copper sulfate in 100 ml of water. First you can like boil it to dissolve it, and then you can cool it, and you can mix the above two. So this will be around 800, this will be 100. After adding of these reagents, some volume may increase. So put it in a volumetric flask and take this what you call as volume to 1 liter. So many of the times you may not require 1 liter, you may need to prepare 100 ml of it. So accordingly, you please tell me that if I want to prepare 100 ml of my Benedict's reagent, how much of these things which I'll add to prepare 100 ml of the reagent? Yes. So I have this Benedict's reagent ready now, right? So after mixing both of these, this is the Benedict's reagent. So to perform the actual test, I'll take around 1 ml of my test solution, the extract or wherever I want to find out ki reducing sugar is present or not. That 1 ml, to that I'll add 2 ml of my Benedict's reagent, and then I'll put it in the boiling water bath. To please keep it in a boiling water bath for 1 or 2 minutes. Like the reaction will take place only if the water is actually boiling. Many of the times people, they don't boil the water and they put it in hot water, then they don't get the result. So please see that your water which is there, it is vigorously boiling. So keep it in boiling water bath. Within few minutes, if you get a color change, a formation of the precipitate of cuprous oxide, then you can say that yes, your sample, it contains what is called as your reducing sugar. So to summarize, Benedict's test is for estimating reducing sugar, uses a Benedict's reagent, formation of a brick red precipitate indicates the presence of the reducing sugar. The color complexes forms, they can be used for quantitatively what you call as analyzing. So I think this much is enough for today. So stay tuned with me, Professor Girish Kukreja, for more in microbiology, biotechnology, biochemistry and much more. Thank you.